Hello guys, in today's lecture we will study protein folding of single domain and multiple domain proteins or multi-domain proteins. In this lecture we will see introduction, what is protein folding, single domain proteins, multiple domain proteins or multi-domain proteins, factors affecting protein folding and its conclusion. So let's begin with introduction. Proteins are essential macromolecules that perform a variety of important functions in a body, from catalyzing chemical reactions to providing structural support. One critical aspect of protein function is their three-dimensional structure, which is determined by a process called as protein folding. In this presentation, we will explore the process of protein folding and its importance in both single and multi-domain proteins. By understanding the basics of protein folding, we can gain insights into how proteins work and how we can manipulate them for therapeutic purposes. So let's see what is protein folding. Protein folding is a process by which a linear chain of amino acids fold into a specific three-dimensional structure or three-dimensional shape. This process is critical for protein function as the shape of protein determines its interactions with other molecules. The process of protein folding is guided by various forces including hydrogen bonds, van der Waals interactions, hydrophobic interactions. These forces act to bring different parts of proteins together, ultimately form a stable functional structure. Here you can see an unfolded protein and here is properly folded protein. So let's see single domain proteins. Single domain proteins are proteins that consist of single continuous chain of amino acids. The process of protein folding in single domain proteins is relatively straightforward, as the protein only need to fold into a single stable structure. However, even small change in the amino acid sequence can have a significant impact on protein folding and function. For example, in mutations, the CFTR gene can lead to misfolding of CFTR protein, which is associated with cystic fibrosis. Here are some examples of single domain proteins like myoglobin. Myoglobin is a single domain protein found in muscle tissues. It is responsible for storing and transporting oxygen to the muscle cells. Like that, lysozyme or ribonuclease A, green fluorescent protein, insulin, ubiquitin. These are some examples of single domain proteins. So now we will see multi-domain proteins. As the name indicates, multi-domain proteins are proteins that consist of multiple domains or distinct regions with their own unique function. In the multiple domain protein, there are multi-domains with multifunction. Many single domains in a single protein that forms multi-domain protein. The process of protein folding in multi-domain proteins is much more complex than a single domain proteins, as each domain must fold correctly and then come together to form stable functional structure. Misfolding of one domain can have ripple effects throughout the entire protein, leading to loss of function or even disease. For example, misfolding of prion protein is associated with neurodegenerative diseases such as Creutzfeldt-Jakob disease. Here are few examples of multi-domain proteins. First is hemoglobin, another is insulin receptor, and the third is DNA polymerase. Here we will see some factors that affect protein folding. Protein folding is affected by temperature, pH, ionic strength, the presence of denaturants. Temperature, pH, and ionic strength can affect the strength of intermolecular forces within a protein, which can ultimately affect its folding. Denaturants can disrupt the hydrophobic interactions and hydrogen bonds that stabilize the protein structure. In conclusion, protein folding is a critical process that determines the structure and function of proteins. Understanding the basics of protein folding in both single and multi-domain proteins can help us to develop new therapies for protein misfolding disease and gain insights into how proteins work. As we continue to explore the complexities of protein folding, we may uncover new strategies for manipulating proteins and designing novel therapeutics. The future of protein folding research is bright and full of potential. Thanks for watching.